go. But I need to do a couple of marketing slides to kind of show you where we play. So a lot of what we do is finishing genomes, because even the human genome was never completely finished. During the Human Genome Project of 10 years ago or whatever, there are huge gaps in the assembly where they couldn't sequence in certain sections of the human genome because the technology doesn't support it. Ours does. And that's because we do really long read lengths. We have the ability to look at base modifications. We can, and I'll show you some slides on that. We can also look at really complex genomic variation, and I'll show you that. Our system is higher accuracy. I'll show you a marketing slide on that one. And we're more sensitive. Because we don't go through this growing or cloning of DNA before you sequence it, because we sequence real time, we don't get the bias that you might get if you extract some DNA, maybe it's got some impurities in it, you clone it and you amplify all those impurities. This is a little bit old, but it shows you of all of the CMWs that sequence, what are the average read lengths of the fragment that you're calling. And at this time, the longest in the 95th percentile, we're up around 23,000 base pair segments that we would get from a given CMW. We've actually seen some of our customers up around 35 to almost 40,000 base pairs long. And this is where our molecular biologists just go crazy, because their whole goal is just shove that as far to the right as possible, because that gives us more and more data off the instrument. And this is an accuracy slide, which compares how, because we have much longer fragments compared to the competition, which has short, like 300, 500, 700 base pair fragments, and we can do, thousands of base pair fragments that, depending on the coverage you've got, we typically will tell customers go for 10 to 15x coverage, that you can then achieve these higher ends of accuracy. But yet I put it all in perspective, 99.999 is 0.001 times, say, 3 billion base pairs of a human genome. So how perfect can you get it, kind of thing. So you have to kind of think through that. So our targeted sequencing, these are different areas where we play, right? Targeted sequencing is, I want to look at this chromosome and I want to go after this segment. I want to really understand what's going on there. Finishing assemblies like the human genome or other really complicated genomes where they're mostly finished, but there's big gaps and they don't really know what the whole picture looks like, the whole puzzle. Our instruments are getting pulled into that a lot. And then also looking at modifications to DNA, and I'll talk about that. So here's an example that was, we got pulled in back in a couple of years ago, and this was the E. coli outbreak in Germany, where they needed to try and characterize this E. coli virus as fast as possible. And we were called in because one of the big things that we can do is there's no amplification of the DNA. You can jump right into it. You can purify the DNA right away. You can get it on our instrument. We can usually call it within 24 hours, and we got involved in that one. And we were successful in helping all the researchers on this one identify 11 different strands of the virus that they were dealing with and help them track down where it was coming from and the types of treatments that you would want to get into. This is another example of a difficult to sequence region. This is a unique example of what we do really well. So in human DNA, and especially like in plants and fish and other really higher genomes, you'll get these repeat regions which can go on forever. In this particular case, I think this is Fragile X. Yeah. So what you've got here is CGG, just goes on, repeat, 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 repeat. You can get these in really long strands. And the researcher we were working with at UC Davis has got one of our instruments. He was able to sequence before and after, and then actually understand what is the distance of this entire region. And that's really important. What he was able to do was identify carriers of autism, mental retardation, Huntington's disease, all these types of things. He was watching this guy was, his wife was a pediatrician, watching the parents bring the children in, and the grandfather's exhibiting symptoms that the wife is not, and the children are potentially susceptible to. And when his wife was telling him about it, he started sequencing them, and he realized he could actually detect these types of diseases. And that's something that it had never been sequenced ever before, because the existing sequencers couldn't do these long repeat regions, because if you can only do 700 base pairs, you don't really know whether you're in the beginning, middle, or the end, or where you started, or how long it really goes.
So if I want to go ahead and study on that, what doctor should I follow?
If you did a search, I'm sorry, if you just did a Google search, find me in the lobby, but I don't know the exact doctor's name. He's come in to talk to us a few times. But he's at UC Davis, and I think if you search Fragile X syndrome, you probably find that paper. I might have it in my reference notes. Mount Sinai is going after the human genome. They're pretty excited about it because, as I mentioned, the human genome has never really been fully sequenced. And they want to go after these regions where they've got these really large gaps, and they know that with our really long fragments, they can actually get through these sequence regions to actually get to it. What's funny is a lot of our customers are saying we're going to get there first, because they're not the only ones that are, some of them are not too loud about what they're doing, but there's a number of our customers. So there's this concept of base modifications, which kind of goes back to, there's 4 nucleotides, A, C, G, and T. And if you think about it, it's like, okay, that's really simple. All DNA is made up of 4 different molecules. How complicated can it be? But what they discovered, and they were researching it until about 20 years ago, that there are modifications where there's molecules like this with the 6-methyl-A, which hangs on to an adenine nucleotide and actually modifies it in such a way that its chemical properties are different. So these are symptoms of ways in which your DNA can go into a tripped up state which causes diseases and things. And being able to see a modified base in your genetic makeup is pretty critical. And so doctors are now getting back into this business because they have this microscope now that can actually let them see it. And the only reason we can see it, and we didn't realize that this is what the technology does, is the enzyme will come along in a normal sequencing segment, and it'll come up next to one of these modified bases, and we see a kinetic signature both before and after, where here's that methyl A, where basically the enzyme's choking or something on it, and we're starting to profile these kinetic signatures and realizing that we can actually start calling these sections within DNA where we're seeing this. And these base modifications are prolific. Once they get involved, they spread in the synthesis process as the genes multiply.
Is that the mechanism in cancer?
It's a precursor to cancer. And this is just some of the stuff we're doing. Just really long, complex molecules. And the big thing is the agriculture. It's really hard to sequence plant genomes because they're very complex and they're very, very long and a ton of repeats. And some of our customers are in the agricultural business, so trying to do that to look at that. That's it.